Hi, this is Mr. Cho, and I'll be going through a few of the practical skills today. So the first practical skill that we're going to start with is just arranging your table properly. Okay, so during the practical, you will have many things, right? For the pure sciences, you just have one set. Right? For the combined sciences, you have two sets. So you need to learn how to organize your stuff properly. Okay, take note of where are some of the common places where your hand will pass through. For example, if you need to use the Bunsen burner, right, your hand will have to go through. So find ways and strategies, right, to move your things that you don't need, right, things that you don't need, try and put them maybe further away, this corner where your hand will not cross through, and things that you need, you can put them right in front of you. Alright, now always start the practical by checking that you have the required material. Right, this will all be listed out on the whiteboard for you, or you'll be listed out on your paper for you. And remember your safety procedure, right? If you're using a Bunsen burner, always remember to wear your safety goggles, and anything that is sharp or breakable, right, or they can break, be careful, right? Don't leave it standing in the tube, and then you might accidentally hit it with your hand. So once you're done with it, put it back into the basket, right? The basket is the safest place where you can put your things. Okay, so that is general idea. Okay, it's very hard. I don't know how to teach you how to organize, but just have in your mind, be organized. I don't want to see anybody spill anything, drop anything, or do that. Now you can ask for additional material, but limited number of times, right? So please don't waste your time, especially if there's liquids, and if you spill it into your paper, you might have to rewrite everything again. Okay, we will still issue you a new piece of paper, but you don't want to waste your time doing that. Okay? Alright, next video. Okay, video 2. So when you are doing any practical, always read through as much of the information as needed. So for example, if right now we are doing 2023 paper, right, I'm going to read through first page, second page, right, third page. I don't need to read the questions yet, maybe just the first few questions, okay, to understand what I'm supposed to find out for, and then I start the paper. Okay, I don't need to read the question 2 yet, right? Question 2 is a completely different question, so don't read that first, right? Just read question 1 and then start question 1, and after that then you can read question 2 and start question 2. Okay, so for example, right now I read, okay, I see that I'm supposed to take warm water, I'm going to take tap water, make a water bath that's 35 degrees Celsius or plus minus 2 degrees Celsius. Okay, in the beaker labeled water bath, right? So I have my beaker labeled water bath, and then I do not want to add more than 100 ml, so I want to keep it little. Okay, now for water, right, tap water, I just get it from my sink. My warm water, I'll get it from my kettle. Usually the kettle is at the side. Okay, if I need more tap water, I can always take. So I'm going to start by mixing. And what do I need for this? I'm going to need my thermometer. Okay, so I start by mixing in some tap water, maybe about a bit. Then I start to mix in the warm water. Okay, I'll give some time for the thermometer to equilibrate. Equilibrate means to get the right temperature. Okay, and again, put all the things that you don't need further away, right, so that you don't accidentally hit them with your hand. Okay, you can always move your things around during the experiments. Okay, make sure that your thermometer, the tip of it, right, the base of it is inside the water, right, so that you can have a proper reading. If it's too little, then it's not good. Okay, so right now I'm about 75. Okay, and my thermometer is a reading of about 35 degrees Celsius. Okay, so that's correct, right? It can be between 35 plus minus 2, so 33 to 37. Okay, now I'm going to take the container labeled A, right, which is here, and I'm going to put it into the water bath. Okay, be careful, don't spill it. Okay, make sure that it doesn't drop out. And then I'm going to cover it with the petri dish over here. This one I don't need anymore. Put it back inside my basket so it don't drop. Okay, so these two right now, warm water, hot water don't need, so put further away. Right, give yourself space. Okay, of course, watch where your things are going. Okay, so now I read the question. It says, okay, so I'll continue with video 3. Okay, so that's video 2. Let's continue with video 3. Alright, video 3. So I'm reading part 1. I read all of the part 1 before I begin the experiment. Okay, don't rush ahead because you have enough time, right? You have 1 hour 15 minutes. You don't want to like do something wrong, then you have to come back to it and do again, then you waste a lot of time. Okay, so I already read it, so now I'm going to start my paper. Okay, it says that I need to wear eye glove and eye protection. Generally, I just wear glove on one hand because I know which hand I'm using, so I just wear that on that hand. Okay, then eye protection. Okay, why? Because although most of these supplements are very safe, but you just want to be sure, right? Some of us may be allergic to yeast, some of us may be allergic to this and that, so just wear. Okay, next, they say that they want me to take a straw, blow gently in the solution in test tube D, right? So I arrange up my test tube already, I already put things now. So you see, I'm arranging my stuff. Things I don't need, I put further. Things that I need, I bring nearer to me. Okay, then this test tube D, right? It contains 3 cm cube of pH indicator solution. Now we know that there are different types of pH indicator solution. Not all of them are just universal indicator. Okay, so we don't really know what is it, but let's find out. Okay, so I take my straw. Okay, and then I start to blow into test tube D. Now, as I'm doing this, my brain should be thinking about what is happening, right? I don't just like mindlessly do it, right? I should be thinking like, what am I doing? Why am I blowing? Okay, so most obviously, when I'm using a straw and blowing, I'm exhaling, right? So you must be linking it to your processes in your body. What process happens to you, or what's the process happening when you're exhaling, right? You're exhaling out what? Gas. Oxygen? No, right? You're exhaling out carbon dioxide. And when is carbon dioxide being produced in your body? Right? It's during the process of respiration, specifically aerobic respiration. Okay, so this one must be gentle, right? Just blow, and every time I blow, I will look into the solution until the pH indicator stops to change color and it remains as yellow or dark yellow. Right, so take a while. Don't blow too hard. If you blow too hard, the solution will splash out and go into your mouth. Of course, it's safe, but don't purposely try to do it. Okay, so it turned yellow already, which is good, but just as I said, continue to blow until it stops changing and remains yellow. Okay, so I'm starting to think already, what am I doing? I'm exhaling out carbon dioxide. Now, carbon dioxide forms a carbonic acid in solution, so therefore, I'm decreasing the pH of this solution, right? So what is this actually? Why did it turn yellow? Because now the solution is acidic, and so it turns yellow. Okay, so I start to see, right? So this one all just put one side. I keep everything again. For now, I don't need this. Put further away. Okay, so I read the question. Okay, why does the pH indicator change color? So very simple. The pH indicator, right? When I exhale, right? I'm exhaling carbon dioxide. Why? Because of aerobic respiration. So the body is exhaling out carbon dioxide. The carbon dioxide forms carbonic acid in the solution. The pH decreases, and that's the reason why the pH indicator changes color from what color? Just original color was green, from green to yellow, indicating an acid. Okay, let's start. Let's continue on to video 4. Right, video 4. So now I'm going to do the steps already. Okay, so I read carefully. Watch out for some extra information that may be at the top or at the end. Okay, so it says that I need to record the time of the stopwatch throughout the investigation. Okay, they will do that later. Okay, now we do the first step. Step 1, right? Always get systematic, right? And then arrange ourselves, right? So here it says label the empty tubes 1 to 5. Alright, so I take my empty tubes, take my marker, and label them. Now, where is a good place to label? It's somewhere that's facing you. You want to label, you can label both sides also, right? So you know which tube is it easily. Okay, but of course, if you don't want to, you can just label one side. Okay, so I have my 5 test tube. Sorry, let me just pause here. Okay, so I arrange myself. Right now, I need my test tubes. Okay, so I will label them 1, 2, 3, 4, 5, which I've already done so. And then, of course, if you want to label both sides, you know, to make it clear to yourself, go ahead and do it. All right. So, where's a good place to label? Of course, higher above this rack, right? So you must be able to see it one clearly, and don't mix them up, right? Always keep them in the same order. All right. Now I want to put the 5 cm syringe. Use the 5 cm syringe, which is this. Okay, make sure that when you see the syringes, you see correctly. So all the syringe have a marking. This is a 5 cm syringe. Or another word for cm is ml, right? So cm cube, ml. 1 cm cube equals to 1 ml. So it's 5. Don't get mixed up, right? There's another syringe that may be different volume, right? So it's a 12 cm cube syringe. Okay, so now I'm going to take the pH solution C and put into each of the test tube 1 to 5. Alright, so this is my solution C. Arrange it, put it in front of myself. Then I start to suck up. Okay, so the first time you pull the syringe, it might be a little bit stiff, don't worry. But just pull it up as much as you can. Then turn the syringe, knock the air bubble out. Okay, then see that you are on 3. Okay, I don't think you'll, if it's like 3.1, 3.2 or 2.9, still okay lah, but try to get it as close as you can to 3. Alright, and put it in. Okay, one more thing to note for the syringe you see that there's this black part, right? So the black part must go all the way down, so the so-called top, right? If you're seeing here, the top of the black part should be where the line is, okay? Because obviously that's where the solution is, so make sure that the black part goes all the way. Okay, so I wasn't very systematic. I missed out one tube, but don't worry, you can always check by looking below. Okay, so now I don't need this. Put it aside. Don't want to spill it. Put this back in the basket. Okay, I remove the container labeled A from the water bath. Okay, so I remove this. Take out my container. Okay, and of course I'm checking, right, that my thermometer, they asked me to measure the temperature again. Okay, so if I see, oh, maybe it's a bit it's below 35 already, right, then I must pour a bit more warm water in. Try and get it back to 35. Okay, or near to 35 plus minus 2. Okay, so now I put container B right inside my water bath. Okay, so why am I doing this? Right, this is to help to ensure that the solution is at the correct temperature. Okay, now if I need to pour the water bath, I have too much water, I can always do that. Just pour it down the sink. Right, just water. Okay, so I measure the temperature of the yeast solution in container A, and then I record it. Alright, so take my thermometer, I wipe it down, need to, and then I measure it. Okay, thermometer is not instant, so make sure that you wait at least 30 seconds, right, for the thermometer to equilibrate and move to the correct temperature. Okay, if you need to, you can raise it up a bit. Okay, so for me, my temperature is 33, right? 33. It's still okay within the range of 35 plus minus 2. Although ideally it should have been a little bit warmer than that, but never mind. Oh no, actually it's not 33, it's 34. Right, good thing I leave it long enough to equilibrate. Okay, so now I'm going to label it in the space of 1.1, okay, which is over here. Temperature of this solution, alright, 34. I take my pen. Okay, so at this kind of, when you need to do pen and like your stuff, right, you need to write, right? So have your own writing space, so that you can, if you're right-handed, put your book somewhere in the correct space. If you're left-handed, put your book right on the correct space, so you can write and you can do at the same time, right? So there's enough space for you. You should have enough space for it. Okay, I don't need this anymore, so I put my thermometer away. I already measured my temperature. Okay, and now I move on to the next step. Okay, I'm going to start the stopwatch. Okay, for stopwatch, there are a few buttons that you must know. Firstly, start on the right side of the stopwatch, stop, and then the reset button is on the left side of the stopwatch, right? Don't press this one too many times. Just press this side. Okay, so I'm going to start the stopwatch and I'm going to leave the stopwatch running throughout the entire investigation. Okay, so I start the stopwatch, boom, start, just put it one side, put it ideally facing you so that you can see it. You can use your Bunsen burner to put it where you can see. Then, why I'm taking this? Okay, then I use a glass rod. Right, the glass rod to stir the yeast suspension in the container level A. Okay, and then we start to see that there's some kind of froth. Okay, why is it frothing? It's because this yeast is a living creature, right? It's a living organism, so it's doing respiration. Okay, we're not sure whether it's doing aerobic or anaerobic, but we know that it's doing some kind of respiration, right? Because the bubbles indicate a certain kind of gas being formed, right? Most likely carbon dioxide gas. Okay, So the froth will form on the top surface of the yeast suspension. We can dispose of this froth that it overflows on the yeast with the spatula. Okay, so what's the spatula? This. Okay, as we start to see, the froth is coming up. Okay, we can actually, if it's too high, now it's not too high, it's still okay. So if that's happening, then just scoop it down, throw it into the sink. Okay, I remove the tubing of the 10 cm syringe, and then, okay, so just be careful here to give a twist so that you don't yank it out and twist it down so it's nicer. Put it aside. Okay, remove the tubing, push the plunger to the bottom of the syringe, right? So you need to push down, and then hold the syringe with the nozzle about halfway into the yeast label and below the froth. We don't want to suck the froth, we just want to suck the solution. Okay, take out 8 cm cube of the yeast, making sure that we keep the nozzle in the solution. Okay, once again, make sure you know where the black line is. Now, for this part, you will start to see that you suck up a lot of air, right? Especially as you suck at the bottom. So it's okay, right? Just measure 8 cm of the solution, right? So sometimes the black line may not be the most accurate anymore, right? What you can do is you can turn it around, although it's going to be a bit messy. Okay, if you see it upside down as 8, you can be quite confident that it's going to be around 8 already. Okay, sorry, this sideways here. Another way you can do is to remove the air bubble, but that tends to be a bit messy, so I try not to do that if I can. Okay, you can shake it up, and then I can try and shake up the air bubbles and then push it down, then just repeat again. But as I said, it's messy. So if you get mess, take your tissue and just wipe down the area, make sure we should get in. Okay, so we have 8 cm. Okay, what am I supposed to do next? I'm supposed to remove the syringe from the yeast solution, hold the barrel with the nozzle pointing upwards. Okay, so I hold the barrel, okay, try and remove any excess liquid, hold the barrel with the yeast pointing upwards. I don't need this for now, put one side, okay, and then I pull the plunger to the 10 cm mark, okay, to introduce some air space. So plunger go down, now it's at the 10 cm mark, okay. Do not press the plunger. Now there's 2 cm of air in the syringe. Keeping the syringe pointing outward, make sure the yeast suspension does not enter the nozzle. Wipe off any yeast suspension from the syringe with the paper towel. Okay, so using this, wipe. Okay, it's going to be quite dirty. You can always ask for more paper towel if you need to. Okay, now replace the tubing tightly onto the nozzle of the syringe. Okay, so give it a twist, make sure that it goes down in. Okay, I'm just put something. Okay, so it says put the free end of the tubing into the test tube 1, and the test tube as shown in figure 1.1. Note the start time and record it in minutes and seconds. Okay, so what I'm going to do is that sometimes I want to make like certain notes. Alright, so I quickly get my pencil case. Okay, usually this part I can use a pencil. Okay, so let's say I want to do some kind of sketching of notes at the side. Alright, I can do it. Let's say I don't want to like mess up my table yet. I just want to do it on the side corner. Alright, so then I start to know, okay, what's the time now? I look at my stopwatch, I can see it says 4 minutes 30 seconds. Alright, so 4 minutes 4:30. Okay, then I start to gently shake the barrel and the test tube until the pH put it inside. Okay, so I put the nozzle inside, and then this solution can stay over here. Okay, so I started at 4:30, okay, which I've written down already, and then alright, so now I'm just doing step 11. Keep the free end of the tubing in the pH indicator solution. Gently shake both the syringe barrel and test tube 1 until the pH solution turns the same color as seen in test tube D in part 1. Okay, so we see that test tube D is our control, right? We can see that the control. Alright, so I need to shake a bit. Shake both of them gently. Okay, and start to see that colors are changing. Okay, and I want it to I'm just going to wait. Alright, no rush. Alright, wait until eventually it starts to turn the same color. Right now I can see that it's a bit yellow. Okay, So as this is happening, right, during this downtime, whenever you got time that you're not doing anything, always be thinking like, what is this experiment trying to test me on? Is it aerobic respiration? Is it anaerobic respiration? What kind of respiration? What is the processes that are happening? Okay, so we see that, oh, there's glucose, there's different things. Once you see the word glucose, you should be thinking of one of either two things. You either think of aerobic respiration, anaerobic respiration, or think about digestive system, right? Then you can start. Okay, we start to see the color is almost there, right? So it's quite yellow, but not as yellow as the control. So it should be a bit more yellow. Let's just wait a while. Okay, nice. Okay, so it has already turned almost, I would say, the same yellowness, right? So test tube 1 is now the same yellowness as the control D. So now I take note of the time. Okay, so on my stopwatch it says 7 minutes 1 second, right? So 7:01. Okay, now remember for minutes and seconds you write just one dot, so it's like 7.01. Okay, where am I? Also time lost track. Okay, so now we can remove the tubing from test tube 1 and place it into test tube 2. Same thing, noting the start time and the ending time. Okay, so you can have a like moments break. So let's say you want to start it at 7:30. Okay, so now continue on. Okay, shake off all this extra liquid which you don't want. Then continue to test tube 2. Okay, so I started at 7:30. Now, actually, you notice I'm not really writing it on my table yet. I'm writing it on just some random site. Okay, as long as I'm able to keep track what is what, I think that's fine. I can always erase it later. The reason why is because I don't want to like dirty up my table so fast. You know, like, I don't want to like commit to everything yet. I want to get the ideas correct first. Then after that, if I realize, oh, I did something wrong, I can always then fill in the correct answer later. Because once you put it into a table, then it's your answer key. Of course, you can write in pencil first and then you can write in pen later lah. Right, then you fill it up later. Okay, remember, anything you want to answer, except for drawing, must be pen. Alright? Okay, I'm going to cut the video here and I'm going to shortcut all the way to the end. And then we will talk again at the end. Okay, see you there. Okay, so now we fast forward to the end. This is the end of the first part. Okay, what do we see here? We have the 5 test tubes, 1, 2, 3, 4, 5. All of them, they have turned yellow, right? And what does that mean? All of them, the pH has decreased. All of them are acidic now. And I already recorded the time. Okay, I recorded at the side of my book, over here, right? It's a bit messy, but it's okay, right? Remember, I'm just doing this part just as like a draft. Okay, so now I'm going to start to copy over my information, and then I'm going to start to calculate. Okay, so we look at, now we have time to think about the table, right? Time and start in minutes and seconds. Okay, how do I represent minutes and seconds? So I write, there's 2 ways you can do it. Firstly, you can write 4 minute MIN and then 30 second S, no SES. No SEC, just S. That's the unit. A lot of people make the mistake of write SEC. That is not a proper unit. You can write that time in minute, or I bet even better, you just put a dot. Okay, not single dot, dot. So it'll be 4 dot. Okay, or what we call it a colon, and then 30. Okay, that's the time at the start, 4.30, and then time at end for the first test tube was 7.01. Alright, so now I need to calculate the length of time. So again, this way is where you need to do some math lah. Okay, please remember 1 minute got 60 seconds. So what's the easiest way to do it? You take your minutes minus the minutes first, then you left over, you convert everything to seconds, then you minus away. Okay, so how I do it is I take my calculator, okay, which I don't have. Okay, never mind. Take my calculator, convert everything into seconds, right? So 7 minutes, 60 times 7 plus that 1 second. Then I minus away the initial time, which is 4 minutes 30 seconds. So I take that 4 times 60 plus 30, and everything all convert to second, and I get the length of time in seconds. And I do this for all 5 test tubes. Once I've done that, then I can find the mean time. Now mean is very similar to the word average, but it's not really the same. But never mind, you can think of it somewhat like that. Basically, you just take all the 5 timing, okay, and divide by 5. Alright, so whatever the number of seconds is, you just take that number, plus show your working, right, inside the table, show the working. All these numbers add together. You want to bracket also can, then divided by 5. Okay, so that's the first part. Okay, so as we start to think about this, we can start to see what's happening already. Okay, so we see the next part is yeast suspension in 10% glucose solution, okay, which is B. Okay, so again, let's take a look at B. Okay, sorry, I made a mistake there. Sorry, let me rephrase. They want you to find the mean time, right? Okay. Now for this question, or rather for this paper, they actually ask you to only circle a few of your time. So in your book, you actually need to go in and circle. Right? Say for example, I want to choose this test tube, this test tube, this test tube. I chose test tube 3, 4, 5. I didn't choose test tube 1 and 2. I need to choose at least 2 of the results. Then I can calculate the mean time. So in that case, then it will just be test tube 3, 4, 5, then divided by 3. Okay, why are we doing this? It's because as you do the experiment, you also will tell, the first few test tubes takes very long. Like it takes like 2 minutes. Okay. Then after that, the last 3 test tubes, they take about 1 minute each, around there plus minus. Okay, what does it mean? It means that it takes some time for all these things to equilibrate, and there is what I call an abnormality here. There's an abnormality in your data. You don't want to choose the abnormal data. You only want to choose the data that are close to each other to prevent your data from being very extreme. Then you get very inaccurate kind of results. Okay, so that's what I did already. Okay, then now I'm going to move on to step 16, which is the next water bath. Okay, now we go and test our water bath. Okay, obviously it's too low. It's been such a long time already. So I want to actually pour out the water bath. Okay, probably in this case now we need more warm water, right? Since the warm water itself was also cool already. So let's pour a bit more warm water. Okay, don't worry. When you pour in all this kind of water, don't need to measure one. You don't need to see how many. Just get a rough feel can already. Okay, I want it to go back to 35. Okay, good. So now it's about 35. Okay, and then I will let it for maybe 1 or 2 minutes. In the meantime, of course, I can take this time to read the question again to double check. Okay, so I now need to realize that, okay, so I need to wash all these test tubes, right? So 1 to 5, I need to wash them. So what do I do with all this solution? Alright, I can simply take them and then I can just pour them down the sink. Okay, carefully, right, you on the tap, don't on too strong, right? Then you just one by one wash it. You actually start to see that it turns blue, right? Quite cool. Okay, why is it blue? Because water is neutral. Okay, and for this particular indicator, it happens to be that blue is the neutral color. Okay, by the way, this is not universal indicator, so don't just assume that green is the neutral color. Okay, so now I have my test tubes washed. Okay, I want to dry my hand. Okay, and then I put them back over here. Keep them in the right order, right? And if they are a bit wet, just tap them onto your paper. I try to remove the water that's inside. Okay, so 1, 2, 3, 4, 5. Alright, stay organized. Now we are going to do the yeast in the 10% glucose solution. I'm going to remember again, okay, which is in B. Alright, so they are saying remove the container labeled B from the water bath, stir it thoroughly, measure the temperature, same thing. Okay, so right now we have the temperature of the yeast in B. Okay, 33. Okay, quite close to the temperature of 34, which I got just now for the A. Okay, good. I'll put this aside. Now I have my B. Alright, so same story, right? I'm going to need to repeat all the steps, right? So everything that I did before, I'm going to repeat. Okay, so this thing, same tube, also I need to go and wash. Alright, I need to remove the tubing from the syringe and wash the syringe thoroughly under running water. Okay, so everything, right, just spurt it out. This one, you can pop open, like that, very cool open, right, and then just wash everything nicely. This syringe also, the tube also, wash it. Okay, just wash nicely. Okay, once you're done, just stuff it back in. Press a few times to clear out any water inside. Okay, then we are ready. Put it back here. Okay, so we're gonna repeat same story, right? What do we do? Read again from the top in case you forget what to do. Right? Don't just assume that you remember and then do it again. Okay, so same thing, right? I've labeled them. I have the 5 cm syringe. Now I'm going to put the pH indicator into all the 5 things. Okay. Now once again, I'm not gonna do everything again because I will just assume that you can visualize what I'm doing. So I'll just cut to the end of the video, and then let's continue to go and discuss what's happening next. Okay. Okay. I think this is video 6. I can't remember already. Alright. So we have done the experiment for both the 15% glucose and the 10% glucose. Now you just think to yourself, why is there a difference in glucose? Why is one 15, why is one 10? Obviously the more glucose, the higher the rate of respiration. Okay. Now in this case, you also think, is it carbon dioxide, is it lactic acid? Not too sure, right? We don't know whether it's aerobic respiration or is it anaerobic respiration. Then we ask ourselves, did I keep these tubes, right? Are these yeast, are they under wrap? Did I cover them for oxygen? I didn't, right? So there's still oxygen, right? So then that might lead me towards more thinking about more aerobic respiration. Okay? I know we didn't learn about yeast, right? But we are able to use what we learned about the human body, right, to apply to the same. These are all other organisms, like the cells, right? They have the same similar processes also. Okay, so now it's time to answer questions. Okay, so question C says, using the results in table 1/1 suggests why the mean should not be calculated for all the results. Okay, so I already told you just now, it's because you don't want to use the extreme or extremely abnormal results in your answer, because that will increase the inaccuracy in your results. This is what you call as the deviation. You increase the deviation in your results. Okay, so then means the range of result will be inaccurate. Okay, so that's your answer. I will go through more in class the exact wording, how to phrase it, all that. For now, I just do a quick summary. Okay, question D. It says about the respiration. Okay, it's very simple. You just take the time in seconds. So 1000 divided by the T in seconds, and then you get round off to 3 SF, and then that is it. Okay. Now what should we be seeing? It says concentration of glucose and then mean rate of respiration. So concentration of glucose is 15 and 10, right? Then mean rate of respiration. Obviously, you should be thinking that the 15% glucose will have a higher rate or higher mean rate of respiration as compared to the 10, because you got more substrate, right? You got more substrate for the cells to use. They are able to perform more of the respiration, and so that's why the mean should be higher. Now, what happens if you somehow you don't see this result? Like, your 10 is actually higher. Okay, now I leave it up to you. I don't want to say too much, but use your brain. Okay. Okay. Next, you draw question E. You need to draw the grid. Okay, and then draw the bar chart. Okay, remember for bar chart, you must have equal spacing, equal width. Okay, it's not a histogram. Histogram can all be side by side, but bar chart must have that spacing. Okay, then explain why is question F. Why is it necessary to record the temperature? Okay, temperature links to what? Optimum temperature. So optimum temperature, the cells have the enzymes, right, performing the respiration. So you need the optimum temperature for the yeast. Okay, if it's too cold, what might happen to it? It becomes the cell become inactive. If it's too hot, then the cells become denatured. Okay, one other error other than any change in temperature in the method of recording the rate of respiration of yeast and state the effect of accuracy of the result. Okay, this one I will go through in class. Okay, next. Okay, next page says student carried out same investigation found that the rate of respiration at 15% glucose higher than 10% glucose. Explain the result. Okay, so they're trying to give you a lot of hint already. There's a hint in this question itself. They're telling you that 15 should be higher than 10. Okay, so why? Okay, so remember we talked about glucose being the substrate. So With more substrate, the rate of respiration should be higher. Okay, then after that they say original investigation, use pitch indicator to determine the rate of respiration. Students wish to repeat the investigation, use a different way to determine the rate of respiration to a higher degree of precision. Okay, so one way to determine the rate of respiration of the suspension, they'll give a higher degree of precision. Okay, now this is quite complicated, a bit difficult, right? So, what is one thing that we did, right? We look at the color, right? We look at the color. And so we are not able to have that precise timing where we can see that, oh, it's actually perfectly yellow. So in this case, one possible way, okay, there's many other ways we also discuss in class. One possible way would be to use a colorimeter to detect the change in color and measure the exact time in which it changed to the exact color. Okay, then aerobic respiration happens in the mitochondria of the yeast. So they give you a big hint. They say aerobic respiration, so boom, but they already know aerobic respiration. Okay, glucose broken down, mitochondria release carbon dioxide, water. Then the student want to investigate the effect of temperature on activity of mitochondria. It has suspension of mitochondria as well as on yeast. You want to use the same procedure, replacing the yeast with one of mitochondria. So what is the suitable control experiment that the student should include in the investigation, and explain why it's essential to include control. Okay, so we did talk about control, so this is the control D. Okay, I will talk more about the reason about this mitochondria in class. Okay, I'll talk about that. Actually I'll talk about everything else in class. So to sum up, alright, when we are doing experiment, always look at what you're doing. Alright, don't panic, don't like overthink things. Actually, it's not very difficult one. This is O level, it's not at A level experiment. So keep things neat, right? Things that you don't need, put one corner where your hands will not cross or accidentally hit. Okay, your pencil box, your paper, put one side at the corner so that you don't get it too wet. Okay, actually just now my paper got wet, a little bit wet already, which I don't like. Okay, then if you can, right, if you want to, there's a towel here, you can try and dry up your table. Just use the power of evaporation. You evaporate very fast, and then you can start to do work. Okay, put things conveniently. So for example, my stopwatch, I put it nicely here so that I can always be seeing it. I don't put it like upside down or whatever. I put it nicely where I can see. Okay, then all the things that I don't need to put at the back. Okay, once I'm done with the experiment, remember that it's more important to think of the answers than to clean up. Clean up can be right at the end. That means that the timing after the exam over already, then you clean up. Okay, so don't worry about cleaning up at all. Just go and look at your answers, make sure you get the correct answers already. If you really done everything, okay, fine. If you want to clean up, fine. Okay, then just start to clear everything, wash everything, and then can do that. Okay, by right, you actually don't even need to clean up because there's a lot of batches. So it's not like the school practical where you must wash everything. Actually, you can just leave everything for the staff, the lab staff to do. Okay, I think that's it. So this is just one experiment. Okay, this whole experiment, let's call it as the yeast aerobic respiration experiment. Okay, concept is very simple. Steps are also actually quite simple, but the theory is where it's going to be important. So make sure that you all know the theory behind aerobic respiration. Okay, with that, Mr. Cho signing out. I will see you all in class. Bye bye.